dtour makes tour-based visual exploration of high-dimensional data effortless through a unified interface with progressively increasing traversal freedom. It opens with a central scatter surrounded by a gallery of keyframe previews. Clicking a preview advances the central scatter. Both the circular slider and mouse scrolling let you scrub smoothly between keyframes. When you need more control, switch to manual mode. Dimension axes appear as draggable handles, letting you control each dimension's influence on the projection. A grand tour mode offers complementary, random exploration of projection space. Importantly, all transitions between modes are smoothly interpolated so that you never lose track of where points went. Let's explore the 4 examples from the paper interactively. Our first 2 examples show how tours gradually reveal structure that no single projection captures. We sweep a single hyperparameter from attraction-only Laplacian eigenmaps to repulsion-dominated t-SNE. Watch how cluster structure progressively emerges. The continuous Laplacian-like layout gradually separates into the distinct clusters visible in the UMAP-like layout, which gets sharpened in t-SNE. Notice this tight cluster sitting among shirts and pullovers, far from the main trouser cluster. Let's select it. All points are short trousers. Now scrub back to Force Atlas 2 and the cluster dissolves, suggesting this cluster is an artifact of repulsive forces, not genuine structure in the data. Yet not everything is artifactual. Boundary points like these boot-looking bags persist at the intersection of footwear and bag clusters across the entire spectrum. Next, we apply the spectral Fisher-Laplacian tour to 346,000 immune cells from a tumor immunology study. This tour orders projections by how well they separate cell types. The first keyframe separates cells along CD4 versus CD8, the fundamental division between helper and cytotoxic T cells. Subsequent frames resolve finer structure like cytotoxicity, activation, and exhaustion. Now we select regulatory T cells and switch to manual mode. Dragging individual axes reveals which markers distinguish subpopulations within the selection. Our next 2 examples shift from revealing structure to interrogating it. We'll now see how dtour can be used to validate a 2D UMAP layout. A standard single-cell workflow embeds PCA output with UMAP, so touring PC pairs lets us check which UMAP structures are genuine. Here we tour 8 PCs of 276,000 cells from a developing mouse brain atlas. Some structures agree. Gastrulation and ectoderm cells form a consistent progression in every PCA keyframe and in UMAP. This is genuine. Others diverge. Choroid plexus cells appear as 2 distant clusters in UMAP, but form a single cohesive cluster throughout the PCA tour. Blood cells are even more striking. UMAP isolates them as a completely disconnected island, but in the PCA tour, they show no comparable separation. This shows how linking a linear and interpretable tour can help validate nonlinear 2D embeddings. Finally, we use a sequential tour to compare 4 text embedding models of increasing recency on 3 million archive papers. The overall topical landscape is remarkably stable across all 4 models, despite a 10-fold range in model size. But watch F2 LLM. It produces visibly tighter subclusters. A 2D color map encoding position in the Spectre 2 frame makes this displaced cluster immediately visible. Selecting these points and scrubbing back reveals that the same papers disperse across the embedding in all 3 encoder-based models. Only the decoder-based F2 LLM clusters them tightly. It turns out these papers are mostly physics education research with a distinct writing style that only F2 LLM clusters together. This illustrates how sequential tours surface model behavioral differences that no single 2D view can reveal.